The Cannibal's Curse, Kuru in the 1940s. 3 decades earlier, high on the Okapa Plateau in Papua New Guinea, grief wore a public face. Women wailed, men cut their hair, and the South Four people practiced a mortuary rite meant to keep the dead close. Transumption, the careful consumption of relatives so their strength would live on in kin. It wasn't cruelty, it was devotion. Then something started to happen in kitchens and gardens where mourning turned to meals. Hands began to tremble, feet lost the ground, laughter burst out at the wrong times and wouldn't stop. Children and women died first, and whole hamlets felt hollowed out. The stakes were human, immediate, and lopsided. The rites centered in the household, and so did the risk. Women prepared the bodies, handled brains and viscera, and fed children. The disease had an eerie arc. Months of stumbling and shaking, odd giggles that masked fear, then swallowing problems, bedbound days, and silence. The elders said ancestors were angry, outsiders whispered about curses, but funerals kept coming and small graves kept appearing. 2 patrol officers wrote reports that reached physicians. In 1957, a young doctor named Carlton Gajdusek arrived with notebooks and questions, and he stayed. He watched, listened, and documented the clinical picture. Ataxia, tremors, bursts of inappropriate laughter, then progressive loss of speech and movement. He tracked who fell ill and where they lived. Meanwhile, anthropologists like Shirley Lindenbaum and Robert Glass mapped social networks, kinship, and the routes grief took through daily life. The pattern made a terrible sense. The practice of honoring the dead was also transmitting something deadly. But what? Bacteria and viruses were the usual suspects. Samples shipped out of the mountains kept coming back without answers. Microscopes didn't show a germ. Culture plates didn't bloom. Still, the disease marched on. To test whether an infectious agent existed at all, Gajdusek did a brutal thing science sometimes requires. He took brain tissue from people who died of Kuru and inoculated chimpanzees. Then he waited. Years. That delay was its own message. This wasn't a quick pathogen. When the chimps developed a similar neurological decline after long incubation, it proved transmission. Kuru wasn't a curse. It was an infectious encephalopathy. The finding didn't end the epidemic. The community did. Colonial authorities had already pressured the Four to stop transumption. Missionaries called it sinful, administrators illegal. As the practice faded in the late 1950s and early 1960s, new childhood cases dried up, but the long tail of incubation meant adults kept getting sick for decades. The last documented case appeared in 2009, a ghost echo from meals shared in the early 1960s. That timeline taught the world prions could hide in a body for years before showing their hand. What nobody knew at the time was the nature of the agent itself. Gajdusek earned a Nobel for demonstrating transmissibility, but not chemistry. Meanwhile, others were puzzling over a sheep disease called scrapie, with similarly weird resistance to UV and ionizing radiation, types of energy that rip through DNA and RNA. If radiation barely dented the agent, maybe the blueprint wasn't nucleic acid. The Kuru story, mapped onto household practice and incubation data, became a human-scale clue in a larger scientific riddle. Ethics ran alongside the science. The language of cannibalism landed like a slur. The Four preferred transumption, a precise word for a ritual of love and obligation. Researchers learned, slowly, to listen to how the community named itself and grieved. Consent and partnership improved over time. The people who'd already lost so much weren't specimens. They were partners in figuring out what had happened and how to prevent it elsewhere. Back in the villages, the disease's signature moments became micro-stories families told with a hard kind of fondness. A mother who'd once carried sweet potatoes up a ridge without pausing could no longer trust her feet on a flat path. A boy's laughter at a cousin's joke turned shaky, then uncontrollable, then thin. The day a favorite song couldn't summon a dance step marked the point. Everyone feared. Decline had started its final run. Meanwhile, something else was happening in labs an ocean away. The chimp experiment had opened the door. Now scientists pushed into the room and started turning over furniture. If not a virus, then what replicated? In scrapie studies, Stanley Prisoner enriched infectivity in fractions dominated by a protease-resistant protein. PRP-2730, and noted that UV and X-ray doses that should have wrecked genetic material didn't stop transmission. Different strains acted different while carrying no obvious genome. The evidence pointed to a shape, not a sequence. For the four, the immediate impact of ending transsumption was concrete. Fewer funerals, more children growing into parents. For the rest of the world, the impact was conceptual. CuIC proved humans weren't exceptions to a class of diseases already known in animals. It drew a line from a kitchen hearth in Okapa to abattoirs in Britain decades later, when bovine spongiform encephalopathy, mad cow disease, would show how industrial shortcuts could feed a prion back into a herd. It warned hospitals that tools used on brain tissue might carry more than stains if cleaned by ordinary rules. In the aftermath, scientists returned to the mountains not to reopen old wounds, but to close loops. No one born after the practice ended developed CuIC. That simple, devastating fact locked the transmission story. It also framed a new fear. If a cultural practice could move a misfolded protein through a community, what other roots existed in our connected world? Here's where the tone shifts. To understand why radiation didn't stop the agent, and why bleach struggled later, we follow the data into the moment. A scientist named a thing nobody wanted to believe in. The next chapter isn't about kitchens or chimps. It's about a word, prion, and a wager that a protein shape could act like a pathogen. The protein that cheats death, prisoners gamble. While that was happening in the mountains, a young neurologist in San Francisco was staring at data that didn't add up. Stanley Prisoner read the CuIC papers and then fell down the scrapey rabbit hole. Sheep with brains that looked like sponges, agents that shrugged off treatments designed to smash genomes. He kept coming back to a strange radiobiology result from Tikva Alper. UV at 250 nanometers and heavy ionizing radiation. Things that crippled DNA and RNA, barely touched infectivity. If a genome wasn't getting hit, maybe there wasn't one to hit. This was the moment everything changed for him. The central dogma said information flows from nucleic acids to proteins. If you want something to replicate, you need a code. Proteins were effectors, not genome. Proposing otherwise was career carbide. Colleagues warned him, "Don't say it out loud," he said it anyway. He coined a word, prion, for proteinaceous infectious particle, and he made a sharp claim in 1982. The essential agent of scrapey is protein. No hidden virus, no bizarre mini-chromosome, just a host protein misfolded into a conformation that can recruit more of itself. The stakes were real. Grant reviewers rolled their eyes. Some journals bounced the papers. In the lab, failures piled up, preparations lost activity, controls misbehaved. But one result wouldn't die. When they fractionated infected brain and chewed away nucleic acids, infectivity remained. When they degraded protein, infectivity fell. They enriched a core fragment, PRP-2730, partially resistant to proteases, enriched for infectivity. The gene they mapped, PRNP, lived in the host, not in some phantom pathogen. It was like pulling the mask off the villain and finding your own face underneath. Meanwhile, radiation and UV controls kept reinforcing the heresy. Dose curves that would have vaporized a viral genome left titers stubbornly high. People argued maybe the agent was shielded by a protein coat that protected a tiny nucleic acid. Prisoner's team countered with independent inactivation methods, hydroxylamine, nucleases, intercalators that damaged nucleic acids without touching proteins. Infectivity didn't care. But when denaturants and proteases mauled protein structure, it dropped. Shape, not sequence, seemed to carry the instructions. Here's where it gets crazy without getting mystical. Strains, different clinical phenotypes and incubation times, popped up even when the agent supposedly lacked a genome. How do you get stable strains if there's no genetic variation? The answer, he argued, lived in conformation. A single amino acid sequence can adopt multiple stable misfolded states, each templating its own architecture. Think of steel paper clips bent into different jigs. Each jig can stamp more to match itself. No letters change, but the fold encodes behavior. The ridicule turned personal. He got nicknames, some not printable. A senior voice told him to stop disgracing virology. But other datasets began to line up. Inherited human prion diseases, mapped to mutations in PRNP, like the D178N you just met in Fatal Familial Insomnia. In animals, scrapie and then bovine spongiform encephalopathy pointed to host genetics and exposure, not a foreign genome. The protein-only hypothesis didn't just fit, it predicted. Action in the lab meant an ugly truth. This material was hard to kill. Protease resistance wasn't absolute, but it was robust. Formaldehyde fixation and routine autoclaving, the backbone of hospital sterilization, left infectivity lurking. That resistance would later haunt operating rooms, but in the 1980s it mostly fueled skepticism. If it's protein, why doesn't it melt like other proteins? The answer was amyloid, a dense, beta sheet-rich structure that packs into fibrils, physically shielding bonds and aligning backbones so tightly that enzymes and heat struggle to get in. Not immortal, just stubborn. What nobody knew at the time was how deep the family tree would grow. Yeast prions, discovered later, showed that self-templating protein conformations were a broader biological trick, sometimes even beneficial. The human cost kept the focus sharp. Sporadic cases with no clear exposure, familial cases with cruel Mendelian edges, iatrogenic clusters from pituitary hormones or dura mater grafts. The variety made more sense once you accepted that the same protein could misfold into distinct templates, cross-seed in some contexts, and crash species barriers when conformations happened to match. In the aftermath of his gamble, the field reorganized around mechanisms. The story pivoted from what genome to what shape, and then to how does that shape spread. Funding thawed, methods improved. New assays began reading out seeding activity instead of hunting imaginary nucleic acids. The Nobel Committee eventually called, not to bless a personality, but to mark a conceptual rewire. Information can live in structure. This decision would haunt and help medicine. Haunt because tools and tissues once considered safe needed new rules. Help because once you frame disease as a templating process, you can imagine breaking it in two ways. Stop seeds from forming or stop seeds from teaching. To see how that lesson works inside a brain, we have to watch a bad fold recruit a good one. Shape as code, how misfolding spreads. To see the code, start with the normal version. PRP, in its everyday form, call it PRPC, sits on neuron surfaces like a polite neighbor. It's flexible, mostly alpha helical, and constantly chatting with the cell's quality control crew. Then a seed arrives. Pre-C-S key, the same amino acids bent into a different architecture, rich in beta sheets. They meet. The seed offers a handshake that isn't a handshake. Contact flips a region, then another. The native protein matches the intruder's pose, and the pose is the message. One conversion doesn't wreck a brain, but the new convert sticks to the seed, extending it into a protofibril. Add more converts and the fiber grows. Beta sheets stack like pages in a damp book. Friction, cellular motion, and enzymes nick the fiber. It breaks. Now you don't have one seed, you have dozens. Each fragment carries the same misfolded template. That's the moment the graph curves upward. Bad origami teaching a class, and it grades itself. Cells aren't passive. They deploy chaperones that refold proteins, proteasomes that chew mistakes, lysosomes that digest junk. Early on, this proteostasis network keeps up. But amyloid fibrils resist proteases, and the fragments dodge through membranes. Meanwhile, the normal PRP is replenished because the gene keeps making it. The misfolded fraction climbs, tips a threshold, and the repair crew falls behind. Neurons don't explode. They fade, lose synapses, and leave holes like sponge marks under a microscope. Here's what nobody saw at first. The strain lives in the fold. PRP with the same sequence can lock into different beta sheet patterns, each with a distinct surface and energy profile. Those surfaces select which incoming PRP molecules can join. That's why clinical pictures differ, and incubation times vary, even without nucleic acids. Conformation is the cookbook, not the letters. But there's a catch, the species barrier. PE sequences differ between species by a handful of amino acids. Those differences shift the shape space of what's easy to template. A sheep-derived seed meets human PRPC, and sometimes the handshake just doesn't fit. Conversion is slow or never happens. Dose and route matter too. A mouthful is not a neurosurgical smear. A high dose can brute force a low probability fit. Direct inoculation into brain leaps past digestive defenses and hits neurons where PRP is dense. Meanwhile, something else is happening along axons. Neurons traffic cargo forward and back. Misfolded seeds catch a ride in vesicles or skim along membrane contact points. Seeding appears in one brain region, then shows up in connected areas in patterns that match wiring maps. It's not a marching army. It's gossip along a phone tree, moving by contact and convenience. You can watch the code in a tube now. In RT-QuIC and PMCA assays, you take a trace amount of seed, often from cerebrospinal fluid, add a flood of recombinant PRP, and agitate. Conversion starts slow, then surges, then plateaus when substrate runs out. Thioflavin dyes light up the beta sheets. The lag time and slope are fingerprints of seeding efficiency. No genomes, just training structure, an early flag that guides care and infection control. Conformation even explains why bleach fails at ordinary strengths. Beta sheets pack tightly, burying vulnerable bonds. Surfaces adhere to steel, especially when dried, turning instrument crevices into seed banks. That's why protocols call for concentrated sodium hydroxide or hypochlorite, plus extended autoclaving at higher temperatures. Enough to denature and hydrolyze, not just sanitize. Hospitals learned this the hard way when standard sterilization left infectivity behind on tools used in neuro cases. Viral hook. The thing you trust to be clean can carry a memory you can't see. Now imagine a single seed landing in a brain where PRNP carries a destabilizing mutation. The energy barrier to misfolding is lower, the native fold breathes wider, and the handshake gets easier. That's familial disease. In sporadic cases, a rare thermal fluctuation or oxidative nick might flip a molecule for a moment. Most flips refold or get cleared. Very rarely, one lines up just right with a neighboring copy and templates. From there, the kinetics, not intent, decide the future. Dose and time carve the path. A small exposure might smolder for years, converting slowly until the fragment cloud reaches critical mass. A massive dose, like contaminated pituitary extracts once given to children, shrinks the fuse. Root sneaks in again. Oral exposures face proteases, stomach acid, and epithelial barriers. Peripherally, seeds may replicate on follicular dendritic cells before nerving in. Intracerebral exposure bypasses all that. Same code, different delivery. Not all misfolded forms talk to each other. Cross-seeding depends on surface compatibility. Some prion strains barely convert across species. Others hop with tweaks. That variability keeps epidemiologists up at night. It also gives drug hunters a lever. Block the key surface, and you block the message. Conformation-specific antibodies, peptidomimetics that cap fibril ends, or small molecules that favor the native fold. Each bets on interrupting the handshake. What nobody knew at the time of the first Kuru transmissions was that soils could archive the code. In chronic wasting disease, prion-laden saliva and carcasses load the environment. Minerals like montmorillonite clay bind PRPSC and paradoxically boost its uptake in animals. The template persists longer than anyone wanted to believe, and every hoofprint is a chance meeting with the seed. The impact of all this isn't theory, it's a timer. Once seeding kinetics pass a point, symptoms accelerate because every new fragment creates more conversion sites.